4.
Okay, so hello everyone. My name is Colin. I'll be presenting with my partner Leon today on the impact of spatial arrangements of passive obstacles on the first passage time of an active system. Our advisor working with us is Professor Momita Disgupta. Random motion is found all around us in nature. The random motion that we are interested in is the protein walking on a DNA strand. A DNA strand is approximately 2 meters long and it needs to fit inside of a nucleus. So a DNA strand needs to form loops in order to be small enough to fit inside of a nucleus. And so here's a short video of the loop formation process of how DNA forms a loop. Uh, Here is a DNA strand indicated in gray, and in red are the passive Crowders. The gold ring here is the protein, and it's moving left to right or in random motion in order to form loops with a DNA strand. Motivated by this system, we are trying to understand the underlying physics of a random walker in the vicinity of a passive Crowder. So, what are we trying to accomplish with the random walker and the passive Crowder? We have two goals. One, we want to know if spatial arrangements of a passive obstacle impact the passage time of a random walker. And two, we want to know if we can arrange the obstacles in different orientations to see if there's a systematic difference in the behavior of the random walker. So in order to understand the system, we must look at the system at a macroscopic scale. So we must define certain objects to the random walker and the passive Crowder. Here you can see the robotic bugs in this image. We use the robotic bugs to represent the random walker, and we cut out small pieces of wood in order to represent the passive Crowder. We can't have the robotic bug move an infinitely long and infinitely wide area, so we must contain the system somehow. And so we made a boundary. Initially, we had a boundary and barrier that are rectangular geometry, but we found that that was one of the key issues that was causing the bug to get stuck very easily. So we changed the geometry of our barrier and our boundary to be a more circular. One key important factor in this experiment is the packing fraction, which you can see is a number of obstacles or barriers in our boundary multiplied by the area of each obstacle divided by the area of the boundary. We found that we can have a packing fraction of up to 6% in order to have the robotic bug not get stuck very easily. Now that we have our experimental setup, we want to characterize our system. We record the bugs moving from one edge of the boundary to another in these two geometries. Once we have at least 10 videos, we convert these videos into TIFF file. What TIFF file does is it takes an image of each frame of the video. With each frame, we can use image J to track the bug's trajectory frame by frame until it makes the first passage. With this, we have the XY positions varying with time of the bug's trajectory, and we can import the bug's trajectory into Python for further analysis. Another key important factor to this system is the mean square displacement, which you can see is 2dt, where d is the diffusion constant and t is time. Alpha is the exponent of t, and for an alpha that is equal to 1, you can see that it's just 2dt, which is just a linear graph, and this is considered a diffusion system. For an alpha that is greater than 1, we would have a super diffusion system, and for an alpha that is less than 1, we have a subdiffusion system. And this is where I'll pass on to my partner Leon to talk more about our experiment.
Hi, everybody. Okay, I'll share my screen. All right, can you see everything where Kong left off? Perfect. On the right, you can see the actual data we've taken. That was with a mix of spherical and rectangular geometry. As you can see, is we graphed it on a logarithmic scale. That is because if you look at the equation on the left, if you take the log of both sides, do some algebra, you can bring the alpha in front of your natural log of t as a constant. And that resembles the equation of a line, meaning as you can see with all our data, it resembles lines. The y-intercept is not important to us. We weren't really interested in that. All we care about is the average slope. And that turns out to be about 1.6, which classifies our system as super diffusive. Being done with the characterization of our data and now knowing how our little robot moves, we went up to our actual experiment. The question we set out to answer what is the impact of different spatial orientations of obstacles on first passage time? In the top right, you can see our setup for the rest of the experiment. We went with the rectangular geometry, and we have two different regions, regions A and B. A is where we put the obstacles. As Kang mentioned, it's always with a packing fraction of 6%, and that doesn't change throughout the entire experiment. Now, all we do is for different ratios of A over B, we record the first passage time. We put our little robot on the left side and let it travel all the way to the right and record the time. And we have some theoretical results from a collaborator. Their system was a bit different since they did a simulation on it. It wasn't two-dimensional, it was one-dimensional. But besides that, the motion and the actual setup of their system is quite similar. They have also a random walking active particle, just in one dimension. And they have regions D where there are obstacles and regions delta where there are no obstacles, which corresponds to our regions A and B. And they found something quite interesting, and for me at least counterintuitive. And that is when D is 20, so the length of the region of an obstacle is 20, you can see that the mean first passage time is biggest when delta is also 20. So when the two regions have a ratio of 1, which at least for me is a bit counterintuitive, because you would think that the more length of obstacle there is, the longer the first passage time would be. But that is not what they found. To relate it back to the video you saw earlier, the mean first passage time is the loop extrusion time. Meaning you saw the little video of the protein forming the loop, and the time it takes to do that is our mean first passage time, just to relate it back to something you saw earlier. And that brings us to the results we have for now. And that is that. We took data at 8 ratios of A over B, starting at 0.4 and going up all the way to 2.8. As you can see, we see the same spike that the simulation showed at a ratio of 1. We have a bit of a difference after a ratio of 1.2. It sort of stays the same. The mean first passage time doesn't increase very much. It stays sort of the same. An important thing to mention is the standard deviation for our system. Normally, you would expect the standard deviation to be very small. That's what you want. You want your experiment to always give you the same answers. But that is just because usually you're dealing with Gaussian distributions. For a system where you measure the mean first passage time, you have an exponential distribution. So a mean first standard deviation of about 50 to 60%, as we have it, is expected. But that also means that you have to take a lot of data points in order to cancel out that standard deviation. For now, we have taken about 30 data points for each of our ratios. So one thing we really want to do is take more of that data to cancel out the standard deviation. And another thing we want to do is to smoothen out the curve. We want to take data at different ratios of A to B. And lastly, in the picture earlier, you saw that for now we have 4 bands of A and 5 bands of B. We want to vary that number in order to see if our system still behaves the same way or is different. And this is an ongoing experiment, so this is what we have planned for now. And lastly, we want to thank our theoretical collaborator, Professor Mithun Kumar Mitra, who did the simulations for us. And we want to thank our department chair, Professor Ben Stortrup, who has been a great support throughout this time and throughout the pandemic. And obviously all my lab partners and Professor Desgupta. Thank you so much. We'll stick around for a couple of questions.
Okay, any question? I think I'm going to start with James' question. So this project was supported with funds for NASA space grant. Why is this project important to NASA?
Khan, do you want me to answer, or Professor Desgupta?
You can go, Leon.
I can give it a try. Well, random motion, or in a sense, just understanding the motion of any active particle on its most fundamental level is quite important for pretty much anything, for any application that comes later on that we might not be able to think of yet, any sort of biological application. It will be important to know how that behaves and how to replicate it on a macroscopic scale.
Yeah. Good answer. Any other question?
So I can kind of, if there's no other question, I can kind of go a little bit more into that. I'm Professor Dasgupta. I am Leon's and Kong's advisor on this project. And yeah, I always feel the whole process of scientific discovery, it's like not linear, and it never comes from like, you know, being exposed to just one sort of things. So I just feel that like, you know, as undergraduates being exposed to a diversity of projects and diversity of things early on in careers of budding scientists, it kind of gives them the experience to handle projects and look at things from different perspectives. So I think that's, as an undergraduate, that's kind of something at Augsburg, we try to encourage our students to get these exposures and through different research experiences, sort of even kind of conceptualizing something which is theoretical and then taking that and creating a project out of it and doing experiments and finding things and realizing that experimentally. It's all a learning process. And I think that kind of gives us some experience in doing that, which would be helpful for as physicists as we progress in our careers.
Oh, thank you so much for this contribution. Okay, so now what I'm going to do, I'm going to stop recording